Hey, hey, hey. We're going to continue with part 2 of our study of macromolecules here, and we're going to pick it up with lipids. Hopefully you enjoyed part 1. Hopefully it wasn't too confusing for you. We're going to take 3 parts to get this done. It's a lot of information in this chapter, so I don't want to shortchange you on any of your information you're trying to get. So if you remember today, we talked about lipids as being fats, oils, and waxes. And our steroids also fall in this category. And we're going to spend primarily most of our time talking about fats and oils. But hopefully you'll get all this. And I hope you like my little cartoon there of Fat Albert. You probably don't even know who that is. But maybe some of you do. Your parents probably do if you don't. Let's talk about fats. They're composed of, there's 2 parts to them. You have your your glycerol. These are your glycerol molecule over here. Okay, and then you have 3 fatty acids. There's one there, and one way over there. And because we have 3 of those fatty acids, we say that those are usually triglycerides or triglycerol, that's the main component. Again, they're formed just like we looked at today by taking the hydrogen and the hydroxyl group off and attaching them in there. So when you take the water out, let's go into some specific types of fats. All right, the first one we're going to talk about are saturated fats. You've all heard of saturated fats. You've always heard diets high in saturated fats contribute to hardening of your arteries and heart disease and things like that. And if you take a look at one of these saturated fats, I'm going to kind of explain to you why we call them saturated fats. So if you look here, you notice that you carbon. All these carbons are bonded together here. And they're all singly bonded together. And every carbon has 2 hydrogens, except this one on the ends have 3. But every other carbon has 2 hydrogens. It's the maximum amount of hydrogens that they could possibly have. So that's why we say it's saturated, because it's saturated with hydrogen. Now, a couple of things about saturated fats. They're solid at room temperature. You think about butter. Butter is solid at room temperature, lard, things like that. And the source of this type of fat are animals. And so if you look, we'll look at the unsaturated fats here real quick, and you'll see the difference between the two. In an unsaturated fat, what do you notice that's different? Something should jump out at you. Right here, right? Does that jump out at you? That is definitely different. Take a look back. If you'd like here. You see that anywhere in the saturated fats? No. So these unsaturated fats will have 1 or more double bonds, okay? Have 1 or more double bonds. And because they have these 2 carbons are double bonded together, notice what they don't have on this side. They don't have the hydrogen. So they are unsaturated. They don't have as many hydrogens as they possibly could have, and it's all due to this double bond here. Little side note, what kind of conformation would this be here around this double bond? Is that trans or is that cis? Cis, right. So a couple other things about unsaturated fats, they are liquid at room temperature. Those are going to be your oils like vegetable oil, canola oil, olive oil, and the source of these are always plants. And these are a little bit better for you. They're not as bad for you as your saturated fats. You still don't want to just probably eat a whole bunch of fatty foods and things like that have been cooked in vegetable oil and all that, but still they're not quite as bad for you as the saturated fats. Now let's take a look at this next thing here. Your book talks about are these trans fats, and you've all heard of trans fats. And if you've seen, I brought this peanut butter jar here. You probably can't see that. If you look at that real close, extreme close up, it says on the label there that it is hydrogenated. One of the ingredients is hydrogenated vegetable oils. So what takes place with these trans fats are they take these in order to prevent. If you think about peanut butter, if you go buy peanut butter from the store and you open up the lid and it's all good to eat, and it's ready to go, and it's solid in there. And what happens is they hydrogenate that peanut butter so that there's a lot of there's peanut oil is in there, and that peanut oil tends to separate out. And so if you look I brought a jar out of this here. Can you see this? This is all natural peanut butter, and you can see that oil in there, hopefully, if I can get in front of the camera. And see how it's separated from the solid. And so what they did with peanut butter is they hydrogenated that, and that's to keep it from that oil from separating out. And when they hydrogenate that unsaturated fats, they're wanting to put more hydrogens on it to kind of make it saturated so they can make it solid at room temperature. And what happened was, when they did this, all these, when they hydrogenate these things, let me pull this up here, they got this right here. So they started getting these double bonds in here. And what kind of formation is that? Conformation is that? Is that cis or trans? You notice how that's trans. They're on opposite side of each other. So as it turns out, trans fats are, like it says down here, they contribute more to cardiovascular disease than saturated fats. So they're really bad. So that's why a lot of things that you eat now will say zero trans fats. So that's a big kicker now. And even now you can buy peanut butter. Like I said, you can buy all natural peanut butter. This is Smucker's. So anyway, and what you just have to do there when you get those, you just have to stir it up, and it's just fine. You stir that oil up into the peanut butter. So that's a little bit about trans fats. So let's go ahead, and one other thing about fats that's very important to talk about are these phospholipids. Okay, we've mentioned this already back in chapter 2 or 3, I can't remember. We talked about how the phospholipids, there's a hydrophobic and a hydrophilic end. So we're not going to spend much time on this for sake of time, but if you notice, this is a main component. These phospholipids are a main component of the cell membrane. So all your cells have these phospholipids that make up the cell membrane that surrounds your cell. And it separates the outside of the cell from the inside of the cell. So if you take a look at it here, it's composed of a hydrophilic head and a hydrophobic tail. And those tail, that's where the lipids come in. You have long chains of carbons and hydrogens here. So those are the fatty acid tail or lipid tail. And they're hydrophobic. So that's a main component of the cell membrane there. So let's go ahead and go talk about the next subject we're going to just kind of introduce with this chapter. I'm not going to get it all done today, but our proteins. Proteins are, like it says here, are instrumental in almost everything organisms do. I always tell my students that when you look in a mirror, you're basically looking at the results of protein synthesis. Your skin, your hair, your muscles, all that is a result of proteins. Your book says that 50% of the dry mass of your cells is protein. And really one of the most important aspects of protein are these things here in the middle, enzymes. You have enzymes play such an important role in their job, just like anything, like it says here, they speed up chemical reactions in your body. They act as a catalyst. You probably learned about catalysts last year in physical science. But I remember catalysts speed up a chemical reaction without being used up. And so that's kind of the way these enzymes work. And you have thousands and thousands of these proteins, these enzymes and different proteins in your body. And they're the most complex molecules known. We'll talk about how their structure of proteins later on in the next video, but they're quite complicated little structures there. I do want to talk a little bit about enzymes, and this will be the last thing we talk about in this presentation. Enzymes are such an important, there's some key things you need to understand about enzymes, and we'll be doing a lab on enzymes coming up hopefully very soon, maybe one day next week. But there's a couple key features to enzymes I want you to notice. Okay, this is a typical enzyme. So you start with enzyme available with enzyme available with empty active site. So this is the active site. This enzyme, this particular one is what we would call is sucrase. Just like we talked about how sugars all end in OSE, enzymes usually end in ASE. So that's kind of a characteristic of enzymes. You'll notice that throughout the year. But they have this active site, and it's kind of like if you think about a lock and key. So this would kind of be like a lock. So the substrate, here's the substrate. For this particular enzyme, the substrate is sucrose. All right, so sucrose is the substrate for the enzyme sucrase. They kind of go hand in hand, the substrate and the enzyme. So what happens is the substrate comes in here and it will bind to that enzyme, and it's caused an induced fit. It's like a lock and key, like we said. We're going to add water right here to the whole process, and when that happens, that enzyme will break the bond between that sucrose. And what you end up with, if you remember, we talked about sucrose is a disaccharide, and so it's going to break that disaccharide into two monosaccharides, glucose and fructose here. So that is an important feat. And then what happens is, once it releases its enzyme, it's a substrate there, it's free to go ahead and bond to another sucrose molecule and break that down again. So it's the way enzymes work, and they're very specific. They don't just work for anything. This sucrase will only work for sucrose. And all these enzymes are very specific to their substrate that they need to act upon. So hopefully you kind of get a little bit of idea how enzymes work. We'll talk more in the next video about, we'll continue with proteins, and then we'll wrap it all up with nucleic acids on this chapter. So hopefully you have learned something, and take this time. I've got a couple minutes here. I'm not going to take it all, but now is a good time to go read your book. I know it's late at night, so you might not want to do it tonight, but you need to read your book. Don't forget that. So, and I will see you guys in class.